So welcome, first of all, to the Life Science Expo. It is great to have everyone here together in this first event. Our panel is called Precision Medicine Innovation, highlighting much of the great work that's going on in the southern New England area in the field of precision medicine. And I thought I'd offer a few comments, and we'll have an interactive panel as we go forward. So one of the challenges with precision medicine, it means something very different to everyone as you think about it. But I thought I'd set the stage a little bit for the panel. And really the focus is thinking back to the concept of when the human genome was first draft sequenced in 2001. And that allowed us to build the catalog of human variation. And that human variation obviously is what makes us unique as individuals, as well as makes us likely to respond differently to new therapies, disease risk, and so forth. And so it's been that evolution since that time that really has seen the rapid advancing in our understanding of human disease. And there's the case of rare diseases, where today, while there are over 7,500 rare diseases, we probably understand the absolute genetic cause of about, I think we were saying around 3,000 of those. And those are rare conditions. Much of what we use in precision medicine today is the hallmark of more common variation. And these are common SNPs and differences in frequencies and patterns of SNPs that lead to, again, different propensity to develop disease, to respond to different therapies. And that's a lot of what we will talk about today as we talk about the field of precision medicine, or making more precise approaches to therapies for individual patients in many respects. Our panelists cross the spectrum of precision medicine. And their bios are in the program, so I'm not going to go into detail. But just to mention from my left and going forward, we have Barrett Brady, who is the founder and CEO of NAPSIS 2.0, the rebirth of an amazing nanopore electronic sequencing technology that he'll talk a little bit about. Then we have Katrina Loomis, who is at Pfizer Global Research, based in Groton as well as in Cambridge, and is really driving the field of precision medicine forward, particularly in disease understanding. And then Bill Martin, who is at Epivax, a very successful Rhode Island company that has partnered with the majority of the top 20 pharmaceutical companies to really help develop new therapies in the area of monoclonal antibodies, and making sure they have optimal epitopes and so forth. So you'll hear more about them. So what I wanted to do was really get our panelists to discuss a little bit more about what they do and how what they do fits into this broad spectrum of precision medicine that our panel is covering today. So we cover disease, we cover technology, and we cover using sequence information to really optimize therapies. So I'll start with Katrina in the middle, and interesting sort of foray into it. I started the pharmacogenomics group at Pfizer 20 years ago, at a time when pharmacogenomics was really not even envisioned, so in 1997. And it was a very heretical concept at the time. We would have amazing arguments about whether Pfizer should be involved in something like this. And it's really great to have Katrina here today, because 20 years later, this is embedded into the fabric of Pfizer. So I want Katrina to just talk a little bit about what she does and why it's so important.
Sure, thanks Patrice, and also thank you for paving the way.
Hold the microphone closer.
Sure. So I've been at Pfizer for 10 years, and I've seen a lot of changes over the 10 years in the field of genomics and applying genomics to drug discovery and development. And I would say a lot of what we do is focused on using human genetic data, other population-based data, and integrating that with other omics data to really try to better understand patients and the complex diseases that we focus on. In my field, I focus mainly on cardiovascular and metabolic disease. And so we use very large scale genetic data and other omics data to try to understand what's driving diseases, how the patients might be different, and really with the aim of identifying the best targets and being able to then take those targets and match them up with patients who are most likely to respond. And that becomes, ideally the precision of medicine approach that we're committed to.
And it really, again, is remarkable because Pfizer, that was the Pfizer of Lipitor, clearly has seen a change in the field of oncology, in the field of metabolic diseases. It's very exciting to see. Bill, to spend a little time talking about Epivax. Obviously, Epivax has shaped the field of precision medicine as the company has grown. Maybe you could give a little bit of a historical perspective on the company and then describe a little bit of what you do, because you really are seminal in the life sciences here in Rhode Island.
Sure. Is this good? So Epivax is maybe that's
Get
a
right
little too
close
close.
to it.
Here we go. Epivax was originally, the technology we used originally developed at Brown, and we spun out of Brown in 1998. So we've been doing this for a while. The core technology of our company is the ability to identify T-cell epitopes within protein sequences. So what's a T-cell epitope? It's a very small portion of a protein that can be seen by the human immune system and engage in activated immune response. So what can you do with a T-cell epitope? Well, the original concept was that we would make vaccines, so Epivax, epitopes and vaccines. And we could look at the genome of a pathogen, a virus, for example, and screen out the T-cell epitopes, repackage those, separating the wheat from the chaff, so to speak, and repackage those as very efficient vaccine antigen. So we started doing that in 1998 with a little grant support. And very, very quickly, we had pharma companies come to us and say, well, if you can find T-cell epitopes in pathogens, can you find them in therapeutic proteins? And we said, well, sure, we can look. And they started to send us therapeutic proteins to look at, and over time we developed not just a screening tool, but also some tools for assessing risk of immune response, so that companies could come to us with their new products, typically monoclonal antibodies, and say, do you think that the human immune system is going to react to this and clear it before it can perform its therapeutic function? And we could render an opinion on that and then go one step further and say, here's how you could fix it. We thought that was going to be sort of the niche for our company, that this would be a rescue technology, a monoclonal that's been worked on for years and invested millions and millions of dollars in, and suddenly you find out that it's immunogenic. It turned out that wasn't the way it worked at all. The technology went immediately right to the beginning of the pipeline. And so what we get is a lot of very early stage screening on hits, and we're one data point within a matrix and choosing which products to bring forward. And so it's all about improving the quality of the pipeline well before any sort of results have been established. How's that?
That's good. And I think it is that when you're bringing that monoclonal antibody into the clinic, one of the things you worry the most about is immunogenicity. And being able to address that before you ever get to the clinic, it sounds like where the focus has been.
Yeah, absolutely.
Which is great. Great. Barrett, you as one of the leaders in DNA sequencing technology have experienced what has really been a remarkable evolution of technology aligning with precision medicine. And it is through DNA sequencing technologies over the last 10 years that we have seen a lot of dramatic progress in identifying disease causation, but also now in the clinic for diagnosing, and maybe spend a little time talking about NAPSIS, the technology that you and your company have developed and where you are today would be great.
So Patrice mentioned the Human Genome Project. So I know it's a very diverse audience. So human genome is 3 billion base pairs in length. That's the haploid genome. Since you have 2 copies of each chromosome, it's really 6 billion, but call it 3 billion. The Human Genome Project costs $3 billion. So people say, oh, it was about $1 for every base in the human genome. And now they talk about the $1,000 genome, So they say, okay, that's a 3 million-fold improvement, right? That's a much better improvement than you would have even at the height of the semiconductor industry when that was scaling with Moore's Law. It's much better than anything else in the past. But what that analysis leaves out is that most of the work, most of the money in the Human Genome Project was not actually spent reading the DNA molecules. It was spent figuring out where in the human genome the DNA that you were looking at was, so that you can then read it. So you'd have some context for where it was. And when people talk about the $1,000 genome now, they do that without the benefit of any context. So what that means is, if there's a disease that is caused by a single letter change, like if you have an A where I have a C, that might mean I have a disease. That's relatively straightforward to see. But if my disease is caused by having 10,000 base pairs that you have, but in my case, they're reversed. So a big piece of the genome kind of moved around. That concept is called a structural variant. And that information is not provided in current sequencing technology because they look at small DNA pieces and they don't know where they are in the genome. So NAPSIS focuses on that question of structural variation. And to work on that, we invented a technology called electronic mapping. So we make these semiconductor-based detectors. There are two scientists in the other room. You can see the instruments and see the semiconductor chips that they use. But DNA molecules flow through those chips, and they're electronically read. And since those molecules are about 1,000 times longer than the molecules that most sequencing reads, you can figure out where you are in the chromosome or in the genome. And structural information or structural variation is now thought to be, it's thought to affect about 20 times more of the genome than single base changes are. And each change that's at the structural level is thought to, on average, be about 50 times more clinically important than a single base change. So I'll pause there because I know we have a lot to cover, but that's what we're focused on.
Yeah, and I think what's amazing about that knowledge is it's still unknown to many of us. I've had my genome sequenced. There's very little information about regions of structural rearrangement because you're using what are called short read technologies, typical of what Illumina provides, a $2 billion sequencing company that didn't exist 10 years ago today. So think about the remarkable revolution that has occurred in technology, but that still needs to continue to really understand the genome and its relationship to disease and as for the fields we study. Maybe Katrina might comment on sequencing as a tool that is used by pharma or other tools and technologies, particularly as Barrett has described.
Yeah, so it's been an exciting decade, I think, in the field of human genetics and also applying human genetics to drug discovery and development. And so there have been a few key advances that really enable us within drug discovery and development to really understand our patients better. And a couple of things that stand out to me are advancements in technology, access to data at large scale, and also patients. And so the three of these things coming together has really given us a big boost and enabled us to do a lot more in understanding patients. The technology I think, has been particularly impressive. When we think what we were doing in the field of human genetics 10 years ago to try to understand the relationship between genetic variation, mutations, or common variation in diseases, we were looking at genetic variation, common genetic variation throughout the genome, from a view at 30,000 feet. And while that was exciting and really told us a lot about the connection between a gene and a disease or disease risk, we were missing a lot. And so over the past several years, as technologies advances and sequencing has advanced, we've been able to look at that genetic architecture and genetic variation with a much finer lens. And what Barrett is now talking about is going sort of to the next level, the structural variation. It's not just about the individual variants, but how they're arranged within the DNA molecule and within the chromosome. That's really important. So This is going to help us get an even more granular view and additional information for us to be able to understand how genes and genetic variation relate to diseases. Once we can make those connections, we have really some key insights that we can use to identify new therapeutic targets, things that we have high confidence in because they're directly links a gene to a disease in humans. And it allows us to start to really do much more finer detailed phenotyping and understanding different subgroups of patients. And so the technology advancements that companies like Napsys and others are making are really crucial for us to be able to do our jobs at getting better and faster and more successful at drug discovery.
And really today, sequencing as a measurement is used for so many different insights into biology, whether it be an RNA measurement, microRNA, structural variations, SNPs. It's really been a remarkable revolution. What I wanted to do is spend a little time just briefly on cancer, because I think today precision medicine lives in the field of cancer. And for those of you who have relatives or have been affected by cancer, the future is very bright, and it is really at a remarkable stage in time. I didn't introduce myself effectively, but again, the company that I have just founded here in Providence, I got tired of commuting to Groton, Cambridge, Worcester, Cambridge again, and said, if I'm going to do it, I'm going to do it here. I've lived in Rhode Island for 23 years and have never worked here. So it is a remarkable time for us in life sciences, and I think we all feel that today. Cancer, what Medley Genomics, my company, is focused on is really understanding, again, at the DNA level, the heterogeneity that exists in a patient's tumor. And that is that a patient's tumor is comprised of many different cell types. And when we take a profile of the tumor, we look at just the most common mutation, somatic mutation, that is potentially responsible for that tumor. But the problem is 2 years later, that tumor comes back in a new form, in a resistant form. And Medley Genomics is focused on using novel analytical tools to really dissect that heterogeneity right the time of diagnosis, and really thinking about how you use that information to then monitor patients. So again, sequencing is fundamental, amazing science, and cancer. I think the comment was that about 70% of current cancer therapies today are being developed with a more precision approach to those cancers, and there is huge investment in cancer. Probably we spend about $100 billion on drugs in the cancer space, and that is just going to continue. It is an amazing reflection, and Lori Glimsher, who is the president and CEO of Dana-Farber, recently highlighted the fact that we have an opportunity, if we can continue to make progress, to see cancer become a chronic disease. And I think that's the hope that we all have, much like for HIV. So when we think about cancer, I want Barrett to sort of add more insight into what he, as a platform company, can do to help us really understand cancer at the individual patient level and how we might see that in the future.
Okay, so genomics affects cancer in two ways. So one is a change in what's called your germline genome, which would mean the genome that you have a copy of in every cell in your body, your sort of normal genome. You might have a mutation in there that makes you much more likely than someone else to have cancer. And a famous example of that is the BRCA1 and BRCA2 genes. But cancer is also itself a genomic disease. So you can also have what's called a somatic mutation, meaning that there's a mutation that has caused your tumor. So what makes a cancer cell is that its genome has been altered. And the way in which it's been altered will determine things like how aggressive it is, what therapies it will respond to. And so there are a couple things. So Patrice covered the notion that even within the tumor, each genome is slightly different, and that heterogeneity might mean that even if anyone knows someone who's died of cancer, they probably initially responded to therapy, and then at some point the tumor developed resistance to that therapy, and then they went downhill very quickly. What's really happening there is that there was always this heterogeneous mix of cells with different genomes, and you selected for the ones that were resistant to the therapy that you gave, and then it became very difficult to treat. And what's interesting is, so if you step way back and you ask, like, why is it that we're so much better at treating infectious disease, like bacterial infections, than we are treating cancer? You might say it's because the DNA of bacteria is so different than the DNA of humans, that even before we knew about DNA, we could find something that would kill the bacteria and not kill the humans in whom they were residing. But the DNA in a cancer cell, on a percentage basis, is actually very similar to the DNA in all of your healthy cells, and that's why historically chemotherapy was so toxic. But it turns out, because the human genome is so much larger than the bacterial genome, if you know how to look, there are actually lots of things that are specific to the cancer genome. And if you can figure out how to target them, then you can, treat patients without side effects. And that's why, over recent years, you've heard of all these targeted cancer therapies. That's what those things are doing. So really what you want is sort of as comprehensive a view as possible, and that will involve single nucleotide changes, it'll involve structural changes. Some of the first structural variants ever found were in cancer genomes, and then characterize the heterogeneity within all of that information. So just to kind of put the problem in context, there are approximately as many nucleotides in a single patient's tumor as there are grains of sand on the planet. So ultimately what you want is to know all of those. But until then, we're sort of sampling more and more of those tumors.
That's good. Great. Bill, Epivax is really embarking on a future where it is an individual patient, an individual therapy matched to the tumor epitopes. Can you describe a little bit of that for us? Because it really is the personalization of therapy at the utmost.
Sure. So we talked a little while ago about developing vaccines with T-cell epitopes. So in that case, when we're looking at infectious disease, we can start with a genome that's very foreign, as Barrett just said. Look at a flu virus. It doesn't look human-like at all, very easy for the immune system to recognize. And then we're looking for epitopes that are going to be broadly applicable, that many, many different people are going to recognize, because we want to make one flu vaccine that will work for everyone. When we start thinking about cancer vaccines, cancer therapy, we have to look at the individual tumor, and we have to look for the mutations in that individual tumor, the relatively small portion that will look non-human, look different than the human genome. And then we also have to select epitopes that are specific for that person's immune system. And it turns out our immune systems are all fairly different. I won't go into detail, but there's lots and lots of variation in our individual immune systems. So an epitope that works for me may not work well for Barrett at all. And we have to find those things in nearly real time, because a cancer patient can't wait months and months and months for us to design a vaccine or test a vaccine. And then we have to do that in a sort of FDA-friendly way, which is to say through a structured process that does not involve any human intervention. So we like to call it a lights out process. So what we're developing in Epivax is that we can take the output from the high-speed sequencing machines, read it into our databases, and then look for those differences. We compare a tumor and a normal sample and look for the differences, screen those differences for the presence of T-cell epitopes, and then using our normal vaccine design tools, put those epitopes together into an efficient package and manufacture and deliver. I say manufacture and deliver like that's the easy part. There's lots and lots of stuff going on there, not my area of expertise, but I don't want to make it sound like it's a trivial exercise. And so that's what we're trying to go on a very practical level. And in some retrospective studies having some success, but we're a long ways from done.
I think what's exciting about it is at the forefront of where the field of, some of the field of oncology is going. And you can see others that are trying individual epitope type vaccine trials right now and beginning to get a readout of that. So it gives you a great sense of really the cutting edge science that is going on here in Providence. What I wanted to do, and I do want to make sure we have time for a few questions, is just ask each of our panelists, myself included, is what is unique about being here and what advantages have we had? And I'll start. My technology spun out of Boston University, and they have a fantastic computer science department. Brown, sorry. What did I say? Boston, sorry. Brown University, sorry. I'm up there all the time. But Brown has a great computer science department, and I'd say one of the top 5 in the country, if not the world. So we've benefited from that a lot. What has been great is the relationship with other people in the community, software design for a logo, website. Everything is about the third the cost of what I used to pay in Cambridge. I've been in Cambridge for the last 10 years. And as a startup, every dollar makes a difference. So it's been fun to find the community here. And I'll just go down the road and, your relationships, why Southern New England? Katrina, you have diversity of Cambridge, but what has been beneficial for you and your company, Barrett?
So I think it's a very exciting time for life sciences in Providence. When I was a medical student, so I was class of 2003 from med school at Brown, and I had gotten a meeting at the time with, it was like the president of the Senate or the Speaker of the House. I didn't know who any of these people were, but I was like, I'm going to go and convince them that life sciences and biotech are going to be an important thing in Rhode Island, and we need to make our move now. And this was when sort of all the major magazines had cover stories like the dawn of the biotech century and stuff like this. So I had this whole speech prepared about how with a strategic location, all this intellectual capital and other advantages, and I got in and I sat down with him and I said, biotechnology. And he said, what is that? And he didn't mean specifically, oh, are you talking about large molecule therapeutics? Or no, he meant I've never heard that word before. And so, whereas you look now, there's all this, I'm on the 195 commission, there's all this great development that's happening. And so I think it's a good time. And I noticed it's a great place for hiring people. So we've, over the years in Napsis, hired about 100 people, and only 20% of them had to relocate to take their job, even though they had very specific jobs, someone had a PhD in how DNA moves through silicon pattern devices. So very, very specific skill sets. People didn't have to move to work in Providence, and often they'll have their last jobs, in Cambridge, and they'll have shorter commutes now, even if they might live north of the border. So I think it's a good place, and I think it's improving rapidly as a place to do life sciences.
Okay, Katrina?
So Pfizer has pretty significant research hubs in both southeastern Connecticut and Cambridge, and what I have seen happen over the last probably 5 to 7 years is that Pfizer's become increasingly engaged in the academic communities in New England, and so we really take advantage of the academic institutions and opportunities for collaboration, both in Cambridge, where we're located very closely to a lot of universities, including MIT. But I have also seen a lot of interaction with researchers in Rhode Island and Providence and Brown. So we draw both on the proximity of the researchers. We get speakers in to give seminars. We also collaborate. In fact, one of my colleagues teaches at Brown about once a month, so there's definitely a lot of interaction with the community. The other thing I know, because I am often on the train between Connecticut and Boston, is that there's a lot of people in Providence getting on the train and commuting to the site in Boston, not just for Pfizer, but the other major industry and pharma companies that are there. So because it's easy to move between cities in New England, I think it's a great area to be in. It really facilitates interaction, which is what one of the things that Pfizer was really interested in and putting a space in Cambridge. So it's been really good and beneficial for us to be up in this area.
I don't miss the train. Bill, do you want to comment?
Sure. I think the small size is sort of helpful to us. We're talking about personal medicine. It's sort of the personal relationships. It's Village Rhode Island, we call it, or not 6 degrees of separation, but 3. We certainly benefit by good relationships with some of the institutions. As we mentioned, we're a spin-out from Brown. We were one of the original grant recipients from the Slater Center. I see Rich here in the audience. We've had a good relationship with Lifespan. Over the years, we've had some very effective and useful banking relationships that were sort of based on personal knowledge and personal commitment. So I think it's the small size. And I grew up here, so for me, it's nice to be home. And I also consider it a very low stress city. And startups are stress enough, so not to have to stress about the city is sort of helpful. So.
I think Bill's comment, Slater, Rich Rand, who is here, plays a key role in this innovation economy, in helping really fund innovative ideas that are high risk and may not be, and really appreciate that seed capital that comes along. So it's another part of the ecosystem. I wanted to make sure we had time for one or two questions, and I can't believe with this audience that there's not one or two questions that broadly that I would welcome for the panelists. Please. I can't believe you're that shy. Okay, I'll ask a question because I'm really intrigued. So 23andMe, how many people have heard of 23andMe? Okay. So 23andMe probably has collected DNA from probably 2 million people by now?
I would say they're probably approaching that now.
Yeah, so let's say 1.8 million. And the majority of those people sign a consent for the use of their DNA for research. And Pfizer has taken advantage of that in what last summer was a remarkable publication in CNS disorders, in looking collectively at a study. And I wanted Katrina just to mention, just briefly, because it is so important, because I never thought it would be valuable. And I was proved wrong.
23andMe has been a great partner for us, and one of the things that I mentioned earlier, one of the incredible changes I've seen over the past decade is really patient engagement. And so many of us are interested in quantifying things about ourselves. I don't know how many people walk around with a Fitbit or a similar device. I've got to watch that tracks my steps, right? So people want to know all about themselves. And 23andMe, the engagement that they have gotten in the sheer numbers of patients that they've gotten to donate their, or to actually purchase their genotyping and consent to research has really been amazing. And then answer all of the questions that they ask so that researchers all over the world can take advantage of understanding the link between genotype and phenotype to gain insights into disease and disease risk, as well as a lot of other non-disease traits like eye color and hair color. But for us, it's been extremely beneficial because people are so engaged with science and research on their own health. 23andMe has been able to provide data to allow us to do things like the major depression GWAS, which was the largest study ever. So it's really the patient engagement and enabling researchers, not just in pharma, but all across industry and academia, to better understand diseases and really make leaps and bounds towards new therapeutics.
Great. Good. Any other questions? Yes. Go ahead, Tracy. Yeah. I think we have time for one or two, right, Tom? Great.
This has been great. You guys do a wonderful job of explaining something that's really complicated.
Barry helped us make sure we did that.
I'm curious to hear you guys talk about cost, right, in personalized medicine. So as we're going away from the older model of developing a huge, expensive drug that's going to be used on a huge population, as we're moving into a capitated system where we're going to be paid for outcomes, how does that factor in?
I'm going to let Barrett take that because he teaches a class in sort of biotech and healthcare, and it's a complicated one.
Okay,
and he's an MD.
Let's play a game. What percentage of healthcare costs in the United States do you think go to prescription drugs?
Give him brackets.
Just someone shout out an answer.
8%, 12%.
You guys only see, that didn't work well because the only people who knew the answer answered. Okay.
Yes.
So I think the most recent estimate I've seen is 9%.
Yes.
And most of the times when you ask people this question, they think it's the majority because it's such a visible expense. And something like 75% of interactions between a patient and a physician result in a prescription being written. And without prescription drugs, most non-surgeons are much more akin to priests than they are to physicians, right? Because that's the primary way in which they intervene. And so a really high percentage of the value of the entire medical system is actually coming from that 9%. But it happens to be a very visible 9% that people pay out of pocket, so they're very sensitive to it. So that's one thing I would say is that the return on investment for prescription drugs is actually pretty good, and we can explain why other countries pay less, but it's not usually the reason people think. And then, on the other hand, the productivity of the pharmaceutical industry has been going down over sort of a long time, and one reason for that is that we're operating under the impression that most medical conditions now have at least one good therapy. But that's not actually true, because once you sort of understand that hypertension is not a homogeneous disease, but it's actually a bunch of different diseases, you realize that those better classified diseases, some of those actually have no good therapies. So there's a sort of a range from like what Bill is talking about, where you're actually using sort of genomic information on an individual level, to somewhere in the middle where you're sort of better stratifying still large patient populations. But I think that if you look at diagnostic spending, right, it's even a fraction of therapeutic spending. So, but it's what will allow you to make the correct diagnosis and give the correct therapy to people. So I think that the return on investment for personalized medicine is incredibly high compared to the rest of the medical system.
I'll give my comment on that. I totally agree with everything you said. From an early discovery research perspective, which is the space that I, for the most part, work in, we have a couple of things that are driving us to precision medicine. If we can better identify the patient subgroup that's going to respond to the therapy we're working on, our clinical trials will be faster and cheaper and more successful. So in the short term, it's good for us. In the longer term, we think that we're going to be in a better position with payers, because if we're more successful, if our drugs are more successful, it'll be easier for them for us to get reimbursement for them. So it's really, it's challenging, because when you first start out, your patient population may be smaller, but at the end of the day, it may prove to be our best way to improve the success of R&D.
Yeah, it is a complicated, but also particularly in rare diseases where you can identify the actual individual with the disease, and the cost of therapy is significant, but the significant benefit to that patient is huge. In rare diseases, it typically takes 8 to 10 years before you have a diagnosis. If you could actually get that diagnosis when that baby's 2 years old and begin to think about that personalization of the therapy for that, whether it be gene therapy or others, it will make a huge difference in the cost of healthcare. All right, I think, Tom, are we done? All right, I'd like to thank our panelists. Give them a round.